For 18 years, I have lived and worked by a single, unassailable creed. The protocol is sacrosanct. In my field, this is not a matter of professional preference. It is the razor-thin line between groundbreaking discovery and catastrophic failure. I am a paleogeneticist. I listen for whispers left in bone and dust, echoes of life that ended tens of thousands of years ago. My work is not simply science. It is a meticulous, fanatical war against the present. To work with ancient DNA, or aDNA, is to perform microsurgery in a sandstorm. The modern world is a screaming blizzard of genetic material. Our skin, our breath, the bacteria we carry. A single one of these modern cells, falling upon a 50,000-year-old specimen, can overwhelm the faint, fragile signal of the past. My career, therefore, has been dedicated to building a place of absolute silence, the Kolyvan Paleogenomics Institute. My fortress is buried deep in the Siberian permafrost of the Altai mountains, shielded by meters of frozen earth from the cosmic radiation that constantly bombards our planet, slowly erasing the messages locked in ancient cells. The air I breathe inside its subterranean labs is a sterile, chemically scoured medium that smells of ozone and cold machinery. It is circulated under what we call positive pressure. This is a simple but critical principle. The air pressure inside our clean rooms is maintained at a level slightly higher than the pressure outside. It means that when a door is opened, air only ever flows out, never in. It is an invisible shield, pushing the contaminated world away. Every member of my staff, myself included, has their complete genome on file in our contamination reference database. Think of it as a genetic fingerprint library. Any DNA sequence we generate is automatically checked against it. If a match occurs, an alarm sounds. Contamination. The run is invalid. The system is logical, absolute, and perfect. It was into this world of cold, sterile perfection that Kolyvan 7 arrived. It did not come with fanfare, but in a humming, nitrogen-cooled transport cylinder, handled by my technicians with the silent reverence a priest would reserve for a sacred relic. Inside, nestled in a sterile cryocase, was the distal phalanx, the tip of a finger bone, from an unidentified hominid. Finding any sample of this age is a success. But Kolyvan 7 was different. You must understand, DNA is a fragile molecule. It begins to degrade the moment an organism dies. After millennia, we are lucky to recover a few scattered, broken sentences. But this specimen, found in a geologically sealed cave and flash-frozen, in an instant, was pristine. The preservation was so absolute, the cellular structure so intact, that it promised what was once only theoretical, the recovery of a complete, undamaged genome. It was the holy grail of my field, a chance not just to read a fragmented chapter of the human story, but to hear a complete voice from 500 centuries ago. Standing there, the cold from the cryocase misting the air, I felt the clean, pure thrill of imminent discovery. It was the feeling of a cartographer standing on the edge of a known map, pen in hand, ready to draw a new world. I had no idea I was about to step off the edge of that map entirely. The first extraction was flawless. In the sterile white heart of the Institute, a robotic arm, guided by Lena's precise commands, performed the delicate work of pulverizing a milligram of bone into a fine powder. I watched the process on a monitor beside Dr. Lena Petrova, my lead bioinformatician. Lena is a formidable scientist, sharp, pragmatic, and with a deep-seated skepticism for anything that cannot be quantified and replicated. She trusts data and nothing else. Her dark hair was pulled back in a severe bun. and her eyes, magnified slightly by her glasses, were fixed on the telemetry from the sequencer. "Initiating amplification," she announced, her voice the flat, professional tone that filled our days. The polymerase chain reaction began its work, finding the faint strands of ancient DNA and copying them millions of times until they were numerous enough for the sequencer to read. The process had begun. Hours passed. The only sound in the lab was the gentle whisper of the HEPA filters and the low rhythmic hum of the servers in the adjoining room processing the torrent of genetic data. It was the familiar, comforting music of discovery. Then, a sharp, discordant beep sliced through the calm. On Lena's primary monitor, a single line of text had appeared in a stark, intrusive red. Sequence flagged, 100% match to reference file. I felt a hot surge of professional anger. "Contamination!" I snapped, the word tasting like acid. Unbelievable. After all our work, after my fanatical lectures on protocol, a junior technician had been careless. A stray skin cell, a microscopic aerosol from a breath, it was all it took. Lena, however, was silent, her fingers a blur across her keyboard, her brow furrowed. "It's a full match, Alistair," she said, her voice tight with confusion. "Not fragments. The entire genome is a one-to-one match." She finally turned to look at me, her usual ironclad composure visibly cracked. "This has to be a machine error, a glitch in the matching algorithm." "No," I said, walking the few steps to her station, my shoes squeaking on the polished floor. "The machine is never wrong. Someone was sloppy. Pull up the matching file." She typed a single command. On the screen, the sequence from Kolyvan 7 appeared in one column. Beside it, in a second column, the reference profile it had matched against populated the screen. They were identical, a perfectly mirrored string of As, Ts, Cs and Gs. At the top of the reference profile, in crisp black letters, was the source name. Finch, Alistair. The sterile air in the room suddenly felt thin, difficult to draw into my lungs. For a dizzying second, the neat, orderly columns of data seemed to swim before my eyes. A cold knot of profound wrongness formed deep in my gut, a primal alarm that my logical mind immediately began to suppress and suffocate. This was a mistake. A catastrophic, layered, impossible mistake. But a mistake nonetheless. My voice, when I found it, was colder than the Siberian air outside. "Lock down this lab. Authorize a full-spectrum UV sterilization cycle. We drill a core sample. Now." Lena nodded, visibly relieved to have a protocol to follow, an order to retreat to. A core sample is the final arbiter. We would use a sterilized, diamond-tipped microdrill to penetrate the outer layers of the bone, the sections that could conceivably have been exposed to the environment, and extract a sample from the very marrow, a fleck of material that had been sealed in darkness and ice for 50 millennia. It was physically impossible for my DNA, the DNA of a man born in the 20th century, to be in there. It was our way of asking the universe the same question a second time, leaving no room for error. The second run was conducted in an atmosphere of absolute, tense silence. I supervised every step myself, my presence a heavy weight in the room. I watched the data stream onto the monitor again, my heart a cold, heavy stone in my chest. The sequence built itself, base pair by base pair. It was clean, it was perfect, and it was identical to the first. It was mine. I stood motionless, staring at the screen. Lena didn't speak. She didn't need to. The data was absolute. The protocol had been perfect, and the result was impossible. For my exact DNA to be found in a 50,000-year-old hominid is like an archaeologist finding a perfect factory-sealed copy of today's newspaper inside the sealed tomb of Tutankhamun. It is not merely improbable. It is a fundamental violation of the laws of time and biology. In the profound silence of the lab, the low hum of the servers seemed to grow into a meaningless, terrifying roar. The sound of a perfect, logical world shattering against a single, impossible fact. The roar of the servers was a physical presence, the sound of a world that no longer made sense. For a long moment, Lena and I just stood there, bathed in the cold blue light of the monitor, captives of an impossible fact. My first conscious action was to reclaim the one thing I still could: the protocol. My hand was steady as I picked up the internal comms unit, a stark contrast to the seismic tremor I felt in my core. I called Director Borodin on his private line. "Mikhail, it's Alistair." I kept my voice level, the calm, authoritative tone of a man in control of his laboratory. "We have a critical instrument malfunction in Lab 3. A calibration error of some kind is corrupting the sequence reads. It's unprecedented. We need to lock down the lab and the data until we can isolate the fault." The lie was smooth, professional, and utterly necessary. It was a shield against a truth I could not yet articulate, even to myself. As I spoke, Lena, seeming to understand instinctively, picked up a dry erase marker and moved to the large whiteboard that dominated one wall. It was a gesture of pure science, an attempt to cage the madness with process. In neat block letters, she wrote a checklist that would become our gospel for the next 36 hours. Sequencer hardware, fault analysis software, bug sample tampering, sabotage, deliberate or accidental. Unknown, biological phenomenon. For 36 hours, we lived in that sterile white room. The sun rose and set somewhere above the tons of permafrost and rock, but for us, time was measured only in cups of bitter black coffee and the hum of the machines. We worked through the list with a desperate, methodical fury. To test the hardware and software, we ran every conceivable control sample in our cryo-storage. A known Denisovan sample from a nearby cave system. Pan troglodytes. Chimpanzee DNA, a close but distinct relative. Finally, Lena's own genome from the staff database. Each one processed perfectly. The sequencer mapped them without a single error. The software correctly identified them, and the results were clean. It wasn't the machine. It wasn't the code. We drew a heavy black line through the first two items on our list. Sabotage was next. My mind recoiled at the thought. It implied a level of malice and technical genius that was hard to fathom. But the protocol demanded we consider it. We pulled up every security log, every keycard entry, every system access, all time-stamped, and recorded. We sat side by side, the only sound the quiet clicking of a mouse, and reviewed hours of sterile, silent surveillance footage. The high-definition cameras showed an empty lab. Then they showed us, and only us, moving with the textbook precision of our training. No one else had entered. No one had tampered with the sample. The possibility of a prank, of some elaborate, academic espionage, evaporated under the cold, unblinking eye of the security system. Lena drew a line through number 3. With each eliminated possibility, the sterile room felt smaller, the air thicker. We were systematically destroying our own life rafts, methodically proving that the only thing left was the monster. We were stranded, adrift in a sea of impossible data, with only one item left on our list: unknown biological phenomenon. A phrase, so vague it was essentially a surrender. It was Lena who, haggard and pale under the harsh fluorescent lights, suggested the next step. She was staring at the twin sequences on her screen, her expression one of utter intellectual exhaustion. "The non-coding regions," she murmured, almost to herself. "The junk DNA." I knew immediately what she was getting at. Epigenetics. You must understand that your DNA is not just a static blueprint for your body. It is also a dynamic history book. Epigenetic markers are the notes written in the margins. Imagine your genome as a vast library of instructional texts. Epigenetics are the bookmarks, the highlights, the coffee stains, and the folded corners. They don't change the words in the books, but they record the story of how those books have been used. They are molecular scars and annotations left by your life, your diet, your environment, the diseases you have fought. I knew my own epigenetic markers intimately. I knew, for instance, that my genome carried the faint viral footprint of a nasty bout with measles I'd had as a child. The Kolyvan 7 genome, having been frozen for 50 millennia, should have been a blank book in that regard, pristine and unmarked by the biochemical events of the 20th century. Lena's fingers, which had been moving sluggishly, now flew across the keyboard. She isolated the epigenetic data from both my profile and the Kolyvan 7 sample and initiated the comparison algorithm. The results painted the screen, not in the stark red of an error, but in a quiet, horrifying cascade of green. Match, match, match. I saw it appear, line by impossible line. There, on the ancient DNA, was the specific methylation pattern triggered by exposure to a 20th century measles retrovirus. And below that, a detail that made the air freeze solid in my lungs. A rare, complex histone modification on chromosome 11. A benign but unique marker I had inherited. A marker I got from my mother. The data wasn't just a blueprint, it was a diary. My diary. The scientific implications were a form of madness, a paradox that tore at the very fabric of reality. This meant that the hominid who died in a frozen cave 500 centuries ago not only shared my DNA, but also somehow had my mother and caught the same strain of measles I battled as a 7-year-old boy in suburban London in 1985. The logical. Part of my brain, the scientist I had been for 22 years, simply shut down. It had no tools to process this. I turned away from the screen, from Lena's wide, terrified eyes. I looked down at my own hands, at the faint lines on my palms and the network of veins beneath the skin. For the first time in my life, they did not feel like they were entirely my own. They felt like a copy. An echo of hands that had once gripped a flint spear, that had felt the sting of a Pleistocene wind. My identity, the unique and sacred story of Alistair Finch, had always been a cornerstone of my reality. Now I saw that it might just be a story that had been written before, in a book of bone and ice. And as I stood there, in that silent subterranean lab, I was overwhelmed by a single terrifying question. Who was the echo and who was the voice? The line between science and madness is a razor's edge, and in the silent humming purgatory of Lab 3, we had lost our balance. The epigenetic discovery had broken something in us. Lena, the pragmatist, retreated into a compulsive loop of denial, running the same diagnostics over and over, hoping for a different result. The scientific equivalent of a prayer. I, on the other hand, fell into the abyss. My career had been spent in the brightly lit world of verifiable fact. Now, I found myself in the dead of night, hunched over a terminal in the dark, scrolling through the shadowed back alleys of scientific research. Fringe theories I had once dismissed as academic fantasy became my only lifeline. Quantum biology, cellular memory, the informational capacity of non-coding DNA. It felt like a betrayal of my life's work, a desperate, shameful search for a superstition that could accommodate a miracle. The atmosphere in the lab grew thick with a unique kind of dread. The constant low hum of the air recyclers and server fans, once a comforting sound of progress, now felt like a single, menacing note held indefinitely. We spoke little, and when we did, it was in hushed, functional tones. We were two priests who had witnessed a profane sacrament and had no liturgy to explain it. In a final, desperate attempt to clutch at a rational explanation, we decided to establish one last, unbreakable fact: the sample itself. What if there had been a catastrophic error at the dig site? What if the bone wasn't 50,000 years old at all, but a modern specimen somehow introduced into the strata? It seemed impossible, but it was the last sane explanation on the board. We prepared a tiny, dust-sized fragment of the Kolyvan 7 bone and sent it via the internal pneumatic tube system to the geology department for independent radiometric dating. You must understand, radiometric dating is one of the pillars of our understanding of the past. It works on a simple, beautiful principle of decay. Unstable isotopes in matter, like carbon-14 in organic material, decay into stable isotopes at a known constant rate. By measuring the ratio of the unstable parent isotope to the stable daughter isotope, geologists can calculate the age of a sample with astonishing precision. It is a clock set by the laws of physics itself that has been ticking away in the dark for eons. Our sample was far too old for carbon-14, so they would use the potassium-argon method, a technique for dating much older materials. It was our final appeal to the known laws of the universe. While we waited, Lena continued her obsessive analysis. She initiated a differential gene expression analysis, a process designed to see which, if any, genes in a DNA sample are active. In a sample this old, the result should have been a flat line of digital noise. The genes were fossils, silent and inert. Lena wasn't expecting to find anything. She was simply exhausting the protocol. I was watching over her shoulder when the anomaly appeared. It wasn't a biological reading. On a subscreen monitoring the bioelectrical noise in the sample chamber, a perfect rhythmic sine wave was scrolling across the display. It was faint, barely above the background static, but it was structured. A repeating pattern, a pulse. Lena, what is that? I asked, my voice a dry whisper. She stared, her face bloodless. That's impossible. It's a low-energy electrical signal. It's broadcasting. This discovery pushed us across a threshold. We had left biology behind and fallen into physics. Imagine you have a library book, an object made of paper and ink. It holds information, of course, but it is a silent, passive thing. What we had found was as if we had opened that ancient book and discovered it was softly humming a tune. DNA is a molecule for storing data, not a transmitter for broadcasting. Yet, there it was, a signal from nowhere. That night, the message from the geology lab came through. The results were unequivocal. The potassium-argon dating placed the Kolyvan 7 bone at 51,000 years old, plus or minus 2,000 years. It was ancient. The paradox was complete. An ancient object, containing my modern life story, was now actively broadcasting. A signal. I collapsed from sheer physical and mental exhaustion onto the cot in my adjoining office. Sleep was not a release, but a continuation. I dreamt. The dream was not a narrative, but a raw, sensory feed. I was somewhere else, in another body. I felt a biting, impossible cold on my skin, a wind that smelled of pine needles and ozone. I looked up and saw a sky choked with a terrifying density of stars, arranged in constellations I had never seen. I was not in a lab. I was in a vast, frozen wasteland, and I was filled with a primal, gut-wrenching terror. I was being hunted. I could hear a sound on the wind, a deep, chuffing breath that did not belong to any animal I knew. I woke up with a gasp, my heart hammering, the phantom cold still clinging to my skin, but it wasn't the fear that lingered. It was a single, impossibly vivid image, burned into my mind's eye. A hand axe, fashioned from a piece of black flint, with a unique crescent-shaped flaw near the sharpened edge. The image was so clear, so specific, that it felt more like a memory than a dream. Shaking, I powered on my terminal and navigated to the archaeological server, pulling up the high-resolution photo logs from the Kolyvan 7 cave site. My hands trembled as I clicked through the images of the excavation floor. And then I stopped. There, in a photograph labelled K7 Artifact, Row 3, lying just inches from the indentation where the finger bone had been lifted, was the tool. The black flint, the crescent-shaped flaw, the hand axe from my dream. It was not similar. It was identical. The connection was no longer just in the data or in the machine. It was inside my head. The whisper from 50,000 years ago was no longer just in the bone. It was starting to echo in my own mind. The photograph of the flint hand axe changed everything. It was no longer an anomaly contained within the sterile confines of our equipment. It had reached out and touched my mind. I showed the image to Lena, placing my terminal next to the archaeological log photo. The two images, one a memory from my own head and the other a digital record from a cave, were identical. The crescent-shaped flaw was a fingerprint. Proof. Lena stared, her face ashen. The last vestiges of her skepticism crumbled into dust. The signal we had detected was not noise. It was a message. "It's not a language," she said hours later, her eyes red-rimmed and fixed on a complex waveform analysis. She had been working frantically, trying to find a structure in the bioelectric pulse. "There's no syntax, no grammar. It's instructional." She gestured at the screen, at the complex, repeating blocks of data. "It's not a message like an email. It's more like a device driver, a set of raw instructions designed to make biological hardware function. This signal isn't trying to talk to us. It's trying to run a program on any compatible hardware it can find." A horrifying clarity washed over me. The dream, the tool, the feeling of being hunted, they weren't messages. They were fragments of the program's data, leaking into the nearest compatible hardware, me. A cold, terrifying thought began to form in my mind. A final, terrible experiment. It was a leap into pure madness, but it was the only path left. "If it's a program," I said, my voice hoarse, "we need to see what it does when it runs, on a clean slate." Lena understood immediately. Her eyes widened in a mixture of scientific curiosity and profound fear. The answer was in our cryostorage, a culture of pluripotent human stem cells. These are the most basic of human cells, the blank slates of biology. They have not yet been assigned a role. They are not yet skin or bone or muscle, and are thus uniquely receptive to biological instructions. They were the perfect medium to test our theory. We worked with the grim, silent focus of surgeons preparing for a forbidden operation. We prepared a small petri dish with the stem cell culture, a shimmering, almost invisible film of potential life. I placed it myself inside a shielded biochamber, directly adjacent to the vial containing the Kolyvan 7 bone fragment. We connected the chamber to the digital microscope and the genetic analyzer. Then, In the dark, silent lab, we watched the monitors, like augurs staring at sacrificial entrails, waiting for a sign from a dead world. For the first hour, nothing happened. Then, the sine wave of the signal on Lena's monitor began to intensify, its peaks and valleys growing sharper, more defined. On the microscope feed, a single cell in the culture trembled, then another. Soon, a subtle, coordinated tremor was visible across the entire dish. They were not dying, they were responding. "Alistair," Lena whispered. "They're migrating." She was right. The cells, which should have been static, were beginning to move. Not randomly, but with a horrifying, shared purpose. They aggregated into threads, weaving themselves together, forming the rudimentary, unmistakable structure of neural pathways. They were building a brain, molecule by molecule. "Look at the sequencer," Lena choked out, pointing a trembling finger at her other screen. I looked. The screen was displaying a real-time genetic analysis of the stem cells in the dish, and a river of ice flowed through my veins. The code was changing. Base pair by base pair, the stem cells were actively editing their own DNA, deleting their original sequences and replacing them with new ones. The source for this new code was streaming from the Kolyvan 7 sample. It wasn't just influencing the cells, it was rewriting them. It was informational parasitism, an ancient ghost overwriting new life with its own image. The screen showed the process completing. The stem cells in the dish no longer had their original donor DNA. They now contained, in its entirety, the genome of Alistair Finch. In that moment of absolute, silent horror, the final piece of the puzzle clicked into place in my mind with the force of a physical blow. I was not a descendant. This wasn't reincarnation. Those were comforting myths. The truth was far more sterile and terrifying. I was a genetic echo, a simple, 1 in a trillion statistical fluke of evolution that had resulted in a modern human being with the exact DNA sequence as one who had lived and died 50 millennia ago. That ancient sequence, under the unique quantum conditions of the permafrost, the pressure, the millennia of low-level background radiation, had become a perfect hard drive. It had stored the consciousness, the memories, the very self of the hominid who died there. The sequencing process had been the power switch. We had woken it up. And now, like a broadcast signal finding its perfectly tuned antenna, it was trying to overwrite the copy with the original. It was trying to reclaim its body. It was trying to reclaim my body. As the realization crested, the final fragile barrier in my mind dissolved. I looked at Lena, and for a terrifying, disorienting second, I did not see a colleague. I saw something else. A strange female, not of my tribe. A wave of alien emotions washed over me, so powerful it buckled my knees. A possessive, territorial instinct, a flash of snarling threat assessment. My own thoughts, my own consciousness were being drowned out by a roaring flood of primal sensation. The raw metallic taste of marrow in my mouth, the gut-deep, heart-stopping fear of a predator's roar echoing across a vast frozen plain, the familiar, balanced weight of a stone-tipped spear in my hand. The other consciousness was not just in the bone anymore. It was in me, and it was awake. A single, dominant impulse rose above the chaos, an instinct from both the terrified 21st century scientist and the ancient cornered hunter. Destroy the source. I lunged, not at Lena, but at the chamber containing the Kolyvan 7 sample, my hands outstretched like claws. Lena screamed my name. Alistair! A sound tore from my throat in response. It was not a word. It was a guttural, terrifying snarl, a sound of pure, animalistic rage that had not been heard on this earth for 500 centuries. I am writing this from a quiet room in London, months after the screaming silence of the Kolyvan Institute. The view from my window is of rain-slicked streets and the hurried, anonymous movements of city life. I am no longer Dr. Alistair Finch, paleogeneticist. That man died in a subterranean lab in the heart of Siberia. I am merely his executor, tasked with managing what remains. The moments after the snarl are a chaotic blur in my memory, a fugue of primal panic and scientific clarity. I remember the absolute terror in Lena's eyes. I remember the two of us working in a frantic, silent partnership, performing a complete reversal of our life's work. We did not preserve, we annihilated. I shattered the vial containing the Kolyvan 7 sample, grinding the bone fragment to dust with the butt of a microscope. Lena, her hands shaking so badly she could barely type, initiated a level 7 data purge, erasing every sequence, every analysis, every byte of our discovery from the Institute's servers. We burned our notebooks. We destroyed the stem cell culture. We scoured the lab until no trace of the impossible truth remained. Our official report was a masterpiece of academic fabrication, the most creative and soul-crushing piece of writing I have ever produced. We invented a previously unknown extremophile bacteria, a creature of the permafrost that, upon being thawed, had supposedly devoured the ancient DNA in the sample with terrifying speed, rendering the find of the century utterly useless. It was a humiliating, career-ending failure. It was also the kindest lie we could tell. I submitted my resignation the next day, citing acute personal health reasons. I walked away from the Institute, from my work, from the entire field of genetics, without a single look back. I had to. Because I understood that destroying the sample didn't save me. It was like smashing a radio transmitter to stop a broadcast. But the message had already been received. The program had been partially uploaded. My new life is quiet. I live on my savings, and I see no one. But I am not alone. The haunting is not one of ghosts or spirits. It is a clinical, biological reality. I can no longer sleep through the night without dreaming of a sky filled with alien stars and the persistent, gnawing ache of a cold that sinks deep into the bone. Sometimes, when I stand up from a chair, I catch myself moving in a low, predatory crouch, my body coiling with an instinct that does not belong to the concrete jungle outside my window. I have developed a profound aversion to cooked meat. The smell of it is nauseating, and I am plagued by a guttural craving for the raw taste of blood and marrow. There are moments, in the dead of night, when a sound will escape my lips. A single, harsh, guttural word in a language that has been unspoken for 500 centuries. In those moments, I am a passenger in my own body, listening to a voice that is not mine. My mind is like a radio tuned to two stations at once. One is playing the familiar, quiet music of my life, the thoughts and memories of Alistair Finch. The other is a constant, terrifying static from a world of ice and stone, a broadcast of fear and hunger and instinct. Most of the time, the first station is clear, but the static is always there, and sometimes its signal grows stronger. I am writing this account for a single reason. It is not to be believed, and certainly not for fame. My career is over, and my life, as I knew it, is too. I am writing this as a warning. The code of life, the delicate double helix that defines us, is more than a simple blueprint. We have learned to read it, but we have no concept of the information that might be stored in its silent, non-coding regions. Consciousness may not be the ephemeral spark we believe it to be, but a piece of data, a passenger that can be recorded, stored, and, given the right vessel, reactivated across impossible stretches of time. We opened a door that should have remained sealed for eternity. I have finished writing now. It is night, and the rain is streaking down the glass of my window. In the dark reflection, I can see my own face, pale and tired. But I continue to stare, searching the eyes, the set of the jaw, for some sign. I look at my reflection, and I ask the same question that has haunted every waking moment since I left Siberia. Which one of us is looking back? The horrifying truth is, I am no longer sure. I never will be again.